Welcome to Lecture 2. Now, I did warn you that we had a lot of complicated and boring terminology, all of it with multiple syllables and Latin and Greek derivations. And what I've tried to do in this lecture in giving you the overview to the tissues and organs is to give you some tools for making the process of, sadly, memorization a little less onerous. So, if you get your outline out, at the end of your outline, you're going to see a table that says analogies. And it'll be helpful if you just have that, have your hand in there. And that'll show you some ways of trying to remember what all of these stupid terms are. The other thing that I'm doing is I'm going to start taking these things and I'm going to try to organize them in such a way that you can make sense out of them. So the first thing I've got after this is a division of labor in the immune system. So here we have the difference between a primary organ and a secondary organ. A primary organ is, not too surprisingly, where things start out. This is where most of the initial cell division that forms these cells takes place. And if we're looking at the adaptive immune system, the primary organs are where the genes rearrange to make all of these exquisitely varied bunches of recognition molecules. And we will, of course, look at that process in detail. A secondary organ is an organ where the immune cells are activated, a counterantigen, talk to each other. So primary organs include the bone marrow for B cells, and most will see myeloid cells, our next distinction, and the thymus for T cells. Secondary organs we will look at again towards the end of this lecture. Those are things like lymph nodes and spleen and regions of the gut. So again, we have these divided into two categories according to their basic functions. Now, the other distinction I gave you again and again, and just as a reminder, we are going to look at the difference between innate and adaptive cells. And the principal difference between these is if they are adaptive, then they have to rearrange genes first and undergo selection. If they're innate, they're there from the get-go. Okay? Now, the last distinction I have to make in this is a little bit tricky. In some ways, I find that my explanations sound kind of circular. But let's go look at it again. Myeloid cells are all going to be innate. The lymphoid cells are the ones that are found mostly in the lymph. Whoops. And the myeloid cells will be found everywhere. The lymphoid cells are found in the lymph. Well, gee, thanks. That's very helpful. Well, this is the way it works. When we look in the next section on the signals that cause cells to differentiate, we will see that one of the primary first decisions that a cell makes as it turns into blood cells is whether to become myeloid or lymphoid. And if it makes the decision to become lymphoid, the cells that it produces will be primarily concentrated in the lymph and in the secondary lymphoid organs at maturity. The lymphoid cells are going to include your B cells and your T cells. So it turns out that everything that is adaptive is also lymphoid. There are, however, some lymphoid cells that are not adaptive, are considered to be innate. And so in addition to the B cells, the T cells, we have NK cells, which we'll look at, and we'll look at all of these in detail later. We also have some of our, what I would call sentinel dendritic cells. and some of those will also be myeloid as well. So we have these kinds of distinctions that I hope will help you to organize your foray into the individual parts of these cells. And I would also like to say I'm about to start showing you huge numbers of figures in the next part, and I have all of those figures on in the figure list, so you don't have to do screen captures. And there you have it. We're going to go into the details. There is one last thing I need to warn you about, and that is the term CD. I refer to this one as the term from hell. The actual CD stands for cluster of differentiation, and that simply refers to how cells come out of various separation procedures that involve a technique called flow cytometry. So basically what happens is you pop the cells in the top of this thing, you flow them one way, you flow them another way, and you wind up with little clusters. And then you name them cluster of differentiation, and then you give it a name 1 on up to, I don't know, it seems like it's heading towards infinity. Okay, so what the word CD means is simply a tag that tells you the order in which it was discovered. Now, it doesn't tell you what it does. It doesn't tell you how big it is. It doesn't tell you what kind of molecule it is overall. It just tells you, and this is somewhat important, is that it's a molecule on the surface of some cell that influenced where it came out during the separation. You see these terms all the time. If you're likely to have heard of anyone, the one you're most likely to have heard of already is CD4, because in HIV, the CD4 plus cells, which are usually TH cells, are the cells that are attacked. And what they're simply saying is that these cells have a protein on the surface that we have identified, as the fourth one discovered. And that protein, which we'll see is used as a co-receptor, will be something that these viruses can attach to. So the sad thing is that, a CD28 molecule is not bigger than a CD4. It was simply discovered later. There aren't categories in these CDs that say which ones are, receptors and which ones are signals. There aren't categories that tell you which ones have immunoglobulin regions and which ones do not. It's simply a first pass through that tells you when it was found, and then the people who found it will give some characteristics of it. So what happens is that once something is labeled CD4, it sometimes gets another label as well, and a common name. And so we will see that there will be places where I say this is CD whatever, and it's also referred to, for example, CD5 is referred to as one of the subunits of the IL-2 receptor. And that can be incredibly confusing, and I apologize for that. If I had been the boss empress of the known universe, I probably would have found a better system, but this is the one you're stuck with, and I'm doing my best to help you deal with it. So with that in mind, we're going to start into looking at the formation of all of these blood cells, and I'm going to try to start using some additional techniques to make it a little more interesting and a little more memorable.